this is not necessarily to be a good prediction of our top gating behavior, because according to this research, again, they are doing dual gate MOSFET, and they're using molybdenum disulfide and aluminum oxide. And according to their research that compare their back and top gating behavior, they realize that the interface condition between the molybdenum disulfide and the silicon dioxide interface have a significant impact on the top gated data when compared to the back gated data. Therefore, again, for our devices, it's helpful for us to see the indium selenide gate characteristic as we showed before. This is a pure indium selenide. So it will be very similar to this setup. So again, they are using dual gate setup, and they have molybdenum disulfide and gallium sulfide. And according to their research, their top gating characteristic have a very high turn-off current and very beautiful gate characteristics. And as for future improvement, I mentioned that about the certain region that is inaccessible for the top gating, but there is a way to get away with it. So in this setup, they use the platinum as their source and drain terminal, and they place the platinum right on top of their substrate, and they use the HBN to pick up the selected tungsten selenide, and then imprint it on the terminal, so that in this way they avoided the region underneath the source and drain terminals. And if we followed a setup like this, we would be very likely to ease our ease our procedures in finding out a good top gating device. And so for now, we don't have a conclusive result for this research, but we do have some indications about the electron transfers between the gallium sulfide and indium selenide flakes. So this is all I have for now. Thank you very much.
Remotely. So you talked about, like, for your devices, you talked about like how, like, you showed how thick were the silicon substrates, but can you tell me, like, what are the thicknesses of the indium selenide and the calcium sulfide?
What are the thicknesses?
Yeah, what are the thicknesses of those devices? Like, are they true two-dimensional materials, or is it like a multi-layer?
They are multi-layers. They are not monolayers. So we are just using mechanical distillation to get the couple of atomic layers of indium selenide and gallium sulfide on top.
So you talked a lot about back gating versus front gating.
Yes.
And I'm not expert in this at all. So I'm curious, do you know which is more common in the field? And is there a really important reason to prefer one versus another, either for physics or for device production?
Well, because top gating has more significance in in industry terms, so they have a more developed, advanced procedures in how to make top gating devices. But for our lab, we make back gating a lot more. Therefore, this is kind of new to us, and that's why we haven't get like the top gating setup right now.
Is there a reason you do back gating a lot more in your lab?
Because this is easier. So for the back gating, we only, as we show in the setup, we have this substrate of silicon dioxide and doped, highly doped silicon underneath. So when we are doing the back gating, we just need to scratch off the surface of silicon oxide and connect it to our probe. So that is an easy step for back gating. But for top gating, we need to use electronic microscope or like photolithography to burn the top metal on the top. So yeah.
I see Professor Lambert has his hand up, so he has a question.
Nice talk. I had a question, maybe you explained it, but could you explain again why you need a different metal for the top gate and for the source and drain? You used palladium for the top gate, right? And nickel, titanium for the source and drain. Do you want both to be ohmic contact, or do you want one to be a Schottky contact, or why do you use palladium for the other one?
Well, this is also the questions I've been asking myself while I was writing the thesis. But in practice, we do use a lot more of the titanium and nickel in order to get a better contact with the p-type semiconductor, because indium selenide is a p-type, so it conduct via electrons rather than the holes. And also for the top gating, it's possibly because it has a layer of gallium sulfide separated, and also I saw a lot of papers that use platinum as top gate rather than other materials. So yeah, I was kind of searching for the question myself as well.
I see. Okay. So it has a higher work function, right? The palladium?
Yes.
Yeah. But in both cases from your pictures, you want to contact with the n-type indium selenide.
What indium selenide?
So in indium selenide, you draw your Fermi level there near the conduction band. So it's n-type, right?
Yes, n-type.
Right. So yeah, okay, I'm still a little puzzled, but that's okay. Why it's different for the top gate and for the source and drain? I mean, they both need to be ohmic contacts, right? Am I not, or am I misunderstanding that?
Yeah.
Yeah, okay. So it must have something to do with that you have this gallium sulfide in between.
Yes.
Okay.
Professor Powell's unmuted himself, so he may have something to say. Shwen?
Wait, let me. Sorry, my speaker doesn't work.
We do hear you.
You hear me?
Yes,
Oh,
I can hear you.
okay. So, yeah, I thought that okay. So, actually, for the gate electrode, it's not for, we don't want to have ohmic contact for the gate. The reason we tried like a palladium is we were thinking to use a different work function metal, different work function metals as a gate electrode to shift the threshold voltage. So to move the, but for the gate, yeah, we don't want to have the ohmic contact.
You want a Schottky contact there. Okay.
Yes.
Okay. Yeah, that's what I was trying to understand. Thank you.
Thank you so much.
Any other questions? And thank you again, then.
Okay, it's a quantum voltage versus temperature cooling down. I get the smooth curve, they burn back up. I get a different curve.
So our next speaker is in the room.
You have to think about where the where the thermal couples are.
And he's not here remotely, and then we have a break scheduled.
Where the
So we may just have a see Professor Cash.
There's a physical difference. Right, so if your sample is down here, but thermocouples up top, they will, the temperatures won't be exactly the same. So like your sample cooling down will always be a little bit warmer than what it says. And then when it's warming back up, your sample will be a little colder than what it says. And that has to do, so it's really like this blue will shift a little bit to the red, and the red will shift a little bit to the left. It's because there's a physical geometric. Oh wow. It's because you're not probing the temperature at the same exact location. Yeah. So if like they go shifted this way, then they should line up. That makes a lot. Okay, thank you so much for that.
So
I thought something really interesting was.
You're using your laptop, I guess you can save me a file.
So again, so the thermocouple will be warmer?
It should be, well, it depends on which direction.
Oh, I see.
Yeah,
So
it had to be in by 9 or I wouldn't
when
see
you're
them.
cooling it, the
So let's see if I
thermocouple
can.
will be the actual temperature, but then your sample will be, it'll say it's colder, but it's actually a little bit warmer than it is. But then when you go back up, it'll be the other way around, where this actual sample will be a little bit colder than we could.
Yeah, thank you so much.
Good job, Dan.
That's fine.
The 3D model.
Oh, so you're asking Brad?
Yeah, so it looks really interesting, right? So when it's warming up, it looks like a completely different graph, right? And this is the same temperature, but really, yeah, you said it's just, it's not a physical phenomenon, it's just shifted over because of the actual location of the
You know, Professor Gass is coming.
She said she's
Cooling down is warmer than
Yeah.
Wait.
Yeah, they didn't tell me that.
Oh, that's gonna be helpful. So I can stand here.
You should stand about here. You can see on my, well, I will share my screen.
Yeah, well, the data analysis actually doesn't seem too hard. Because it's just right? I mean, I don't know how I'm gonna get error bars, because that's a requirement.
Wait, is this the wall?
Yeah.
Share screen.
Are you on the experiment now?
No, this is my first lab report
Yeah,
that I haven't turned in.
Oh, but you're still doing analysis?
Yeah, so the
So you
thing
can see
is,
where you're standing now, that's you. So if you stand about here, you'll be
yeah, so
visible.
we got our achievement center.
And this is the remote tool if you want to use it. Okay. So we're going to start our next talk, the final one before the break. Tongxi. Go ahead.
All right, so hi everyone, I'm Tongxi. My project mentor is Professor Kathleen Cash. Today my topic is going to be analysis of electron recombination in gallium nitride, indium gallium nitride quantum wells using photoluminescence spectroscopy. So just get started. Gallium nitride, indium gallium nitride is a commonly used commercial semiconductor used to make LED light bulbs, which usually emit blue light. So this is the sample I use. It's a quantum well 78. It has a C sulfide substrate and gallium nitride quantum barrier with the indium gallium nitride quantum well. And this one on the right is a transmission electron microscopy of quantum well 78. So indium gallium nitride is a crystalline semiconductor. It releases photon during recombination process. It has a low recombination rate due to polarization. So you can see from the graph, due to polarization, the lowest energy and of the conduction band, the highest energy of the valence band isn't plane. It has some angle, so the wave function tends to locate at the lowest energy of the conduction band, the highest energy of the valence band. So the wave function tends to locate at the edge of the quantum well instead of in the center. So they will have a low overlapping, which means a very low efficiency. To fix this situation, we have an zinc germanium nitride inserted into a quantum well. As you can see from the graph, with the zinc germanium nitride inserted, the highest energy of the valence band shifts to right. So which means the wave function of electron will also shift to right, and the overlapping between the two wave function will increase. Therefore, we will have a higher internal quantum efficiency. So I've mentioned the idea of recombination already. I'm going to explain it more explicitly. Radiative recombination and non-radiative recombination. Radiative recombination just means recombination that emits photon as energy. Non-radiative recombination is a recombination emits energy in the form of heat, vibration, or phonons. Radiative recombination include intrinsic recombination and zinc defect recombination in the quantum well 78. Only in this sample, it will have some zinc defect recombination. So the small peak is the intrinsic recombination, and the large peak is zinc defect recombination. And I will explain why there are this recombination later. And intrinsic recombination is temperature independent. Defect recombination is temperature dependent. That is why at high temperature, will have low, at high temperature will have more non-radiactive recombination, and at low temperature we won't have, we'll have less non-radiactive recombination. Why is intrinsic recombination temperature independent? So intrinsic recombination occurs at the quantum well. Electron falls from conduction band to valence band directly, according to Arrhenius equation. Here, K stands for the reaction rate, A is a parameter, K is R is constant, T is temperature, E is just activation energy. So it won't have any energy barrier, therefore no activation energy. So rate of reaction will be constant. But for defect recombination, it will first need to fall to the defect level, which means it has some energy barrier. So it has a activation energy, therefore it will be temperature dependent. So internal quantum efficiency, just means the ratio of the number of photon emitted to the number of electron hole pairs generated. In this case, it means the number of photon emitted by the quantum well sample to the power of the laser beam. So non-radiative recombination only reaches phonon. This is important because we can calculate the internal quantum efficiency, then we will know how much energy is lost by non-radiative recombination through heat or vibration. This is a model of 3-channel recombination. As I've mentioned before, there's 3-channel. First one, D means defect recombination. I stands for intrinsic recombination. Z in here stands for defect recombination. So capital M is total number of electrons. Change of electron by time equals the Ki means the power of the laser beam minus by the rate of change through defect recombination and intrinsic recombination, also zinc defect recombination. So as it is in steady state, the change of total electron over time will be equal to 0, which means the power of laser beam will equal to the rate of intrinsic recombination, defect recombination, plus the zinc defect recombination. To calculate the quantum efficiency, for example, to calculate the intrinsic recombination, we use rate of intrinsic recombination over total power of the laser beam. By the same method, we can calculate all the radiative recombination, which means the zinc defect recombination plus the intrinsic recombination and the non-radiative recombination. We just simply change the numerator to the defect recombination rate. This apparatus I use a laser beam with a power with a wavelength around 325 nanometer, and this tube filter here is used to fill out some spontaneous emission by laser plasma. There is a neutral density filter which only allows wavelengths of 325 nanometer wavelength to pass through. UV mirror is used to bring the light to the height of the sample. Also, as I'm measuring it through the change of temperature, I need to control temperature by using liquid nitrogen gas and a heater. So I need to control liquid nitrogen gas flow and the power of heater to make the temperature stable. This is a trial that measures the intensity of light with a change of laser beam intensity. As you can see from this graph, when the laser beam intensity increases, the intensity of the light will also increase as expected. But at high temperatures, at high power of laser beam, you can see the intensity of light kind of saturated. So back to my topic, spectrum taken at a constant power but different temperatures. It's kind of hard to tell, but you can see that with the temperature decrease, the intensity of the whole spectrum increase. And also at very low temperature, especially this green one and orange one, you can see there's two peaks. This way is intrinsic recombination. So at low temperature, there's less non-radiative recombination. So the intrinsic recombination will be more apparent. So how to prove the small peak is intrinsic recombination and large peak is zinc defect recombination? I use a Gaussian fitting of the two peaks to identify the position of the intrinsic recombination and zinc defect recombination. Basically, the position of the small peak and large peak. The small peak is located around 4,995 angstrom, and large peak is around 5,500 angstrom. By knowing the angstrom, we can calculate band gap. So the small peak band gap, which is indium gallium nitride band gap, is 2.48 electrovolts, and large peak band gap, zinc defect band gap, is 2.17 electrovolts. The difference between these two band gap is around 0.31 electrovolts. So in previous study by Nakamura's research, he found that the band gap difference between zinc defect and indium gallium nitride band gap is around 0.4 electrovolts to 0.5 electrovolts. But I got 0.31 here. It could be some experimental error, or it just growth condition is different. So the last one is a model fitting for all radioactive recombinations. From this graph, at lowest temperature, the quantum efficiency is 1, which means there's no non-radiactive recombination. So I assume this. I assume at the lowest temperature, there's no non-radiactive recombination to fit this model. And with the temperature increase, in this model, you can see the quantum efficiency decrease. So that's it. Is there any questions?
Questions either in the room or remotely? I'll try a few. One of your early slides, maybe the first one, you showed a picture of your sample. But I don't remember if you told us where you didn't make the sample, right?
I don't make it.
Do you know who made it and how they made it?
I don't know how made it. I don't know how they made it, but I think it's people from Ohio State University.
Okay. I guess another one. You did the fitting with Gaussian peaks, you said, right?
Yeah.
And I think I can imagine why, but I just want to know why you chose Gaussians instead of some other function.
I chose Gaussian simply just because it looks like Gaussian. That's why I chose it, and the fit turns out to be like quite good, so I just keep using it.
Well, the Gaussians usually imply there's something random about the underlying system. Is that what you expect? That there are little random peaks in some region?
No. I won't say about the zinc defect. So the most of the graph I showed with one broad peak is zinc defect recombination, and one broad peak just simply looks like Gaussian. I don't know if it's coincidence or is something related. So I just use Gaussian to fit all these peaks.
Okay. Other questions? Okay. Then it's time for a break. I'll see everybody back here at 3:10 and Wayne. You want to get something? You're going to use your computer? No, we're not ready to start.
Yeah.
Let me just get this Mike, is your question? Sorry, is your question?
I just wanted to say thanks for everything. I got hopefully see you next Thursday. Right? Yeah.
Well, you definitely, if you're coming, I'm coming next Thursday.
Yeah.
You've been very convincing.
No, I mean, oh yes. Yeah.
But definitely next Thursday. You're going to dinner, right?
Yeah.
Dinner.
Okay. Well, let's go.
We're making sure we have fish just for you. A couple vegetarian things just for me.
Nice.
Mentioned you had a question about my presentation. I just wanted to make sure that I'm clear.
Nothing critical. I always try to have questions for everybody. I know what it was. I took the notes. I was just curious, well, how do you model? I can imagine different ways to model a surface. The simplest way would be like a 2D array. Each element gives you a height above the surface. There must be much more sophisticated ways of doing that. I was really being modeled the surface.
That's actually some of what I did. Well, I originally have a 2D by 2D noise type, just Gaussian random white noise. And then I did a Fourier transform, and then so the with the 2D array, and each value was kx, ky.
Okay, so each value is really a height of the surface.
It was the frequency component, and then what I did is I put in the locations of the rays,
Mhm.
and it calculates based off the Fourier series
Okay.
what the height of that ray should be. So it was a 2D array in that sense, but it's also I have it so you can plug in any xy, and because the surface tiles indefinitely far out, as long as you know what the bin size is, you could plug in any xy, and they'll tell you the height of the surface there, and then where the ray, which direction it comes off in.
Because I was wondering, when you start taking derivatives and things, if you have discrete values, you have to maybe fits the spline, all sorts of crazy things. A lot of modern software just does surfaces with other techniques, vectors and so on. So I was just curious.
Now, I didn't actually have to do splines or anything like that because I had enough points that I could, if I just know the rays I want to put in, I can calculate it. I don't have to interpolate, which is the purpose of doing it the way.
So it was just, sorry, it wasn't a critical question.
Oh, okay. I just wanted to make sure. I didn't-
No, I was just curious. Thank you. And of course, you're going to be surprised. This was the Saturday before commencement when you go to the award ceremony.
No, I did see that. I was, I think I was CC'd on.
No, I
that.
did that on purpose
Oh, okay.
because I thought that you were invited to the one, I assume. The other.
I got invited today. They hilariously emailed me from the undergrad studies office and were like, could you please come pick up your award? You want to have the continuing student ceremony? And I was like,
Yeah, but they didn't invite you to the ceremony?
I never got the invite for the ceremony. And
Okay, I was
then-
afraid you'd gotten that, and so whenever I went there, and I said, oh, she's like, so that's why I sent the email, copied you, so
Oh,
you wouldn't
okay.
be too confused about why.
Okay, they didn't invite me, but then I did get the email saying that I wasn't there and to come pick it up. And I was like, are you sure this is meant for one person? And then
Yeah,
they
so
just didn't.
you may want to have them hold off until the actual one, if you want to go, if your parents will be in town or something, and you can go
I
off.
told them I'll be attending, yeah.
Okay, good.
Yeah, so hopefully that's worked out. Thank you.
Yeah, I'll be back way before you start.
Are you done now?
Oh.
Done for the semester? No, I've got an econ exam of all things and a lab report. I was supposed to do a lot of work this weekend, and all I did was research, which I guess was very productive, considering I got literally all of the results you saw in the last 48 hours. I didn't get anything.
That was me trying. I tried to do that the last 48 hours. It was not.
No, over the weekend, I probably did like 20 hours of work over 2.5 days. And so I got 15% of the work done in like 1% of the time of the project. But
Everything everything all right?
Professor Brown?
Oh, Kate, can you hear me?
Yes, I can hear you.
Oh, great. Do you have an idea about what you want to do now? So I don't have to bother you with another email
Oh, yeah.
in the fall?
I am going to ASU's astrophysics program for the PhD.
That's great. An astrophysics program and PhD, where?
Arizona State University.
Oh, great. Oh, that's a great school. Okay, wonderful. Okay, good. I'm all set.
Okay. Oh, I realized I forgot to respond to that email. Thank you for.
No, that's okay. I'm all set.
Okay.
Thank you. Thank you very much.
I wasn't sure if I was coming back here first to finish up my econ degree or just leaving, and I was supposed to take a gap year, but gap year's not happening, so. Scientific algorithm. And so listening to your presentation, I'm sorry I can't stay. I have a meeting to run off to. Oh, I have to wait to be done. I'm gonna turn this to loud.
There we go. Oh, what happened? Oh. Hmm? Oh, fun.
Sorry, not gonna speak for yours.
It's okay. That's pretty much most of what you're Ferrari.
I figured that's what everyone's.
Yeah.
Yep.
See you.
Tables have things. And there's, the food will be left over in an hour. Maybe the students will take it home. Sure, you can put lemonade in here, but I'm not sure much of it will get.
Not much of them here either. But I'll leave it.
Okay,
I'll stick one of the tables down.
thanks. Professor Ryan is going to join us. Give her another couple minutes. Just in case she's going to join us after. I noticed I made a mistake in the schedule. I only gave him 10 minutes. Nobody told me. So which would come back and it won't work for you. Okay. This won't work for you. You're on your own. But you can see here, you'll be visible to the remote audience as long as you stand sort of along here somewhere. That would be okay, or just back here.
Okay.
But if you stand back there, you'll disappear.
Oh. It got really silent in here.
Hmm?
It got really silent in here. It got really silent in here.
Yeah, this often happens towards the end of meetings. People are tired. I'm sorry. Somebody, I'm here the whole time. On the bright side, whoever's left in the room at the end can take home all the extra food.
That is true. For sure, I was like one of the only people who had the coffee.
Coffee, yes. Yeah. Ellen wanted to order 2 things of coffee, and I said, and the tea. Would you have had tea? No, I don't think Winston's. So Gerald, you didn't tell me. I only allotted you 10 minutes for your talk.
Oh, yeah, I was gonna say something about that.
That
but
was
I figured-
that was a mistake.
Yeah, I figured, yeah.
I generally drink a lot of green tea and coffee.
So Mr. Milne, we'll start you whether or not your advisor shows up. We'll let you start on the dot at 3:10.
You just talk really fast.
Is your advisor going to be joining the Zoom? I know Knowles or Christians, Professor Taylor said he's in the faculty senate meeting and he'll try, he's zooming into that and he'll zoom into this too at the same time. Okay. Okay, so it's 3:10, so
Oh, okay.
you're on.
Okay. Hi, everyone. Thanks for being here, even though it's so late. This semester, or this year really, I've been working with Professor Drobitskaya in the math department on a project with machine learning for magnetic resonance fingerprinting. And so to sort of give like an introduction, medical imaging plays a really vital role in disease detection, diagnosis, and treatment monitoring. And this makes a lot of sense because if you're a clinician and you wanted to see how the patient's doing, if having an image just makes it that much easier to evaluate the condition of the patient's body. And so with this medical imaging, there are many techniques to do this, such as MRI, CT, radiomics, and sort of underbellying all these techniques is this more general problem of recovering an image, reconstructing an image from data. And so what's different from all these techniques are these 3 points. So what kind of data are you interested in? How exactly do you map your data back into an image, to what precision and to what accuracy? And how exactly efficient is this data collection process? How much information do you retain and how much noise will there be? And so magnetic resonance in general, the data type that you're interested in are signals. So how it works is that you have a lot of protons in your body and you apply this external magnetic field. And then that aligns most of your protons into the same net direction, as you can see in the orange figure in the Z direction. Then you apply radio frequency pulses, which perturb the protons into 180 or 90 degrees. And then you wait and you measure back the reactant signals, which is essentially the movements of the protons back to their original position. And this is the data that you put through a computer. There's many different smaller variations of the MRI, but essentially you put it through a computer, and then that gives you parameters of which clinicians then view those images. And so how MRF works is that it greatly speeds up the process of MRI. So in MRI, when you're measuring the signals due to the protons, you're looking at position by position basis. MRF works by frame basis. And you do this by greatly undersampling the images that you evaluate of your objects, and then over long periods of time. And then in considering an entire frame as well, you're able to measure multiple parameters. And so the reason why you can take this undersampled frames and map that back into an image, into parameters, is because it's predicted, it's in theory, that every single position from biological factors will give you a unique signal that corresponds to what exactly you're looking at. And so hence the name magnetic resonance fingerprinting. And so these signals are then tabulated and then compared against this table which we refer to as this dictionary. This dictionary is calculated from the block equations from T1 and T2 parameters, which are essentially the longitudinal and latitudinal directions to which these protons go back to their original position, and then to some sort of signal. These signals can be real complex values. And all right, so in more detail, how exactly does MRF work? What is this procedure? And so how we want to consider MRF is really essentially this lossy procedure. When you sample your image, you are applying this a very special kernel. But let me just go through this by one. So here in this example, those of you in the senior project class have seen this cat written many times. You have this cat. And then in this cat, we assign the red and green values as your T1 and T2 values. Then we brought this cat into this Fourier domain. This is your Fourier cat. And then you apply the sampling scheme. The sampling scheme essentially means that you're taking points only along the white. And so you do a piecewise multiplication, piecewise matrix multiplication, and then you obtain essentially figure 3. You take the inverse Fourier transform of it, and you get your lossy cat. This lossy cat is then your signals, to which you then compare to your dictionary. And so that's essentially how the mapping works. So how efficient really is this process? Here, this figure is brought back up again. To the left, it depicts this, the dots depict the points that you actually take, and on the right is this depicting that you take many frames, you compile those together. And so when you take these signals, because you're undersampling them so much, they just appear as noise. And then what's typically done by clinicians is employing a dot product matching scheme, which essentially is an exhaustive search through the dictionary of your signal, and you compare and you find the best match by, let's say, mean-square error. And so this is a little unrealistic when you have that much noise, and the dictionary is as well an ideal picture of what exactly these signals would look like. In addition, when you pre-compute this dictionary, you're putting a limit to how refined your image can eventually be. And so some other solutions, some novel solutions that people have been using are machine learning methods, as well as low-rank approximations. And so the machine learning methods that we're particularly interested in for this research are neural networks. And so these neural networks are really powerful in creating these nonlinear maps, because as depicted here, these networks, which is the blue, the G inverse that we're interested from the measure signals back to your T1 and T2 parameters, are really compositions of functions. And these functions can be linear or nonlinear, and through superimposing the two, you can create functions that go from signals of, let's say, 6,000 back to 2 parameters. And so The problem with neural networks in the research is that researchers have a tendency to over-parameterize, put too many nodes, put too many layers, without really considering that there is some underlying invariance to the design or to the MR problem itself. In addition, low-rank approximation methods are also used in comparison to neural networks where we can take directly the input data to the output data. Lastly, I almost forgot to mention is that these training sets that these researchers use are typically very only medical images. And when you train selectively on medical images, such as like 99% of healthy patient data, 1% of like people with the tumor, then it's very easy to misclassify what you're actually looking at. And so in our project here, we're aiming to create a model, which means not only to design a network, but also to consider the training set to which we are solving this MR problem. And so neural networks, even though they're powerful, you can't really use them to solve every single kind of problem out there, every single kind of mapping. And so what we want to first demonstrate is that the MR problem is indeed solvable, or neural networks is indeed a valid method to approximate this mapping. And so some first formal proof of concept tests were with simple classification and regression networks, where we see that we put in the dictionary entries, and then we output, we predict the T1 and T2 classes and values. And so we see both in both cases, the classification and regression, even with a small subset of the dictionary. The dictionary is a table of unique mappings from T1 and T2 parameters to your signals, we are able to achieve high accuracy. So what does this really mean? This means that even though the dictionary is a unique entity, there are redundancies which allows us to predict T1 and T2 values, even though the network only sees a small subset of them. In addition, from the high scoring of the regressions that we see over 99%, we are able to go beyond the pre-computed refinement limit of just having a dictionary, and we can have further for a much greater precision to the images that you're generating again. And so this is just like a first glance interpolation of the dictionary attempt, where we took only half the values of the dictionary, and then then we tested it on the entirely different subset of the dictionary, and we can see we can get pretty good measurements and guesses for the new ones. And so this really shows us how we can create these super resolutions of dictionaries that eventually that we just see as possible. And so some other results are some really some other methods are that researchers propose that networks have the geometry behind the networks very closely mimics the SVD. More specifically, that the first layer of the matrices mimic the singular values chosen from the low-rank SVD decomposition of matrices. So here we just compare the two using these weight maps. From the left side is the rank 10 approximation of the SVD matrix, which are the U columns, as well as the weight maps for the neural network layers. And we can see here that qualitatively, you can see that they're pretty different. And then we also put from a least squares regression, the distance, the degree difference between the two vector spaces. And as we can see, the numbers are pretty big, they're in degrees. And so that's really showing that this neural network is solving and approaching the MR problem in a totally different way than other low rank approximations, namely the SVD here. And so this begs the question, what exactly is the network doing? What features is it really selecting? And so, which I'll go to the results. And so as of right now, we are still in the works of creating a general training set. So as I mentioned earlier, in the clinical practice, these training sets usually comprise of medical images. We're looking to solve this problem in a more general sense to then achieve greater accuracy by considering the problem as one of parameter recovery from long time sequences. And so, as you can see here, we use many different kinds of images, and we're in the process of trying to converge and trying to achieve a low mean square error value, or you can say a high score. And these scores are essentially the accuracies to the T1 and T2 values that we're trying to obtain. So here is this cat again. This is the true T1 values and predicted T1 values you can see that they're pretty close. The accuracy here is actually around 90%, where the goal is to achieve greater than 99%. Here's another picture of another example of how this image is reconstructed into what accuracy. And then this is where I took T1, the RGB values, and put that through the network and see what we recovered. Because it's only 90% accuracy, it's a little bit of a fuzzy cat, but we can see that we're on our way there in order to be able to create this mapping of the network. And really what we're aiming to do is just add more pictures, add more different kinds of pictures, and eventually obtain a high accuracy. And so while I mentioned earlier that the MRF neural networks solves the MRF problem in a totally different way than low rank approximations. Why is that? And so we're going to be looking into what exactly the invariants are into the problem. And as well, we are making good headway on seeing how should we represent the signals and classes of the time sequence, as well as T1, T2 values. And so here, because the signals are both real and imaginary, there are different ways that you can sort of feed them into the network. So this is sort of a preliminary glance to what exactly that we perceive to be going in the right way. And so as of right now, we're really just testing on the reals and then imaginaries as seen in the first row, since that has seemed to give us the best results so far. And for class representations, not depicted on here, there are different ways that you can label these signals, whether or not it's normalized from 0 to 1, or you can have them weighted by their true values. That is still much in the works. We're trying to see which is the best. And so the conclusions from work I've done this semester so far is that these proof of concept tests were performed to verify the suitability of such modeling for the problems and to great accuracy, to great success. And from these results, we are looking to apply a more general training set. The encoding is very efficient and is a totally different form from SVD, which again shows that we're solving the problem in a different way. And so that is, and some future plans are to really analyze the weight images. The weight images are how, what features in this vector space that the neural network is creating, what's most important. Then we want to optimize our networks. If we can then see what's most important, we want to put images of that type in and then raise our accuracy. Eventually testing on patient data, and then formalizing our results into a sampling theorem is the ultimate goal. And so thank you, Professor Derbyskaya, so much for this semester, and as well as Professor Brown, Professor Martin for looking over the external communities, talks, and Professor Chottiner as well. And thanks for listening. Yeah.
I have a question. So actually, I have two questions. First, if you can go back, in your sampling approach, you have that like spiral.
Yeah.
So can you explain like where you get that spiral and why is that like a good scheme?
Yeah, so there, as you can tell, well, this sampling scheme is only one of many ways you can sample from the from your object. Some other examples would be radially, Cartesian. There are even some people using like different squiggles, but it's just like from the literature search, this is just what has performed the best. So yeah.
Okay, and I have another question. In your neural network, in your output layer,
okay.
What actually are you recording? What actually values do you get in that output layer?
So we normalize in our in actual practice, we normalize the T1 and T2 values to be from 0 to 1. And so you can imagine those, since we're putting in general images, we're putting in a traditional training set images, natural images. They are going to correspond to, let's say, your red layer, your green layer, your blue layer. And so let's say your T1 is your red and your T2 is your green. So just that's hopefully that gives like a physical interpretation of your value, because you're going to be reading for medical imaging or like parameter maps. So
all right, thank you. Interesting presentation.
thanks, Errol.
Other questions?
Yes, so Wei, you're really working on the two hot topics in signal process. I presume you're using a commercial software. for your networks.
Oh,
Is that right?
actually, no. We originally started with MATLAB, but MATLAB's deep learning toolbox is quite outdated as well as the documentation. So we just moved on to Python. So far, we've been testing on Scikit, which is an open source framework. We probably will move to Keras later on, which is still open source, just a Python library to be able to find more fine-tune things. But yeah, we're not using commercial products. So
now I know in talking with you before that you haven't moved to clinical data. What data did you use for T1 and T2? What is this from again? I forget.
So before we're moving on to clinical data, the data that I'm putting in right now, there are the two kinds of testing that I've done in this project so far, which is the dictionary alone and then the general training set. For the dictionary alone, we have the signals, which is the fifth dictionary. But for the general training set, like I said before, because we're looking to solve this in a more general sense, we're using like the red and green values of the picture.
So you're using essentially block equation solutions for T1 and T2.
Yeah.
And for your dictionary. And as I say, one of the great challenges, and it'll be fun to see how it all works out, is, as you know, when Errol was asking you about the sparse, case-based coverage of each of those, this rotating spiral, you get tremendous aliasing, and aliasing is an error that's proportional to the amplitude itself. So it's not Gaussian, it's not random. And it'll be fun to see how, the neural networks may actually, correlate that despite that correlated aliasing error.
Yeah, I mean, after that external committee meeting, I looked into it more, and it seems like some images I didn't put on here, but if you really shrink the layer size you have, and then raise the regularization parameter, where regularization really is a factor that you change that affects how general the network is training to be, which is a little bit more detailed. Anyways, the point that I wanted to share with you is that because the neural networks are bringing your signals, which are high dimensional, into a lower dimensional state, so let's say signals from 6,000 to 100, the distortion that you see from aliasing just sort of like becomes really general, and your image that you output is a lot smoother. And so that's sort of the very glossy explanation.
Oh, I'm hopeful. Yeah, that's great. And the only other thing I'll mention is there's a lot of people doing what you're doing, and you'll want to, there's just gobs and gobs of paper using AI and fingerprinting on all kinds of problems, and it'll be important to kind of be sure we're aware of them. But it'll be fun to talk with you more. Thank you for a good job.
Thanks, Professor Brown.
Other questions? I have one more question. You sort of answered it in your last slide.
Okay.
You said you weren't ready for patient data yet.
Okay.
Because I had an ACL, MCL sprain a decade ago at the end of my basketball career.
Okay.
And the orthopedic surgeon couldn't tell if it was sprained or completely torn. I was wondering if you could solve that for me. But
Like, give me an image eventually, maybe.
he wanted to do surgery, but he said I wouldn't be able to walk by the time I was 60 unless I loved him. But he also said he got a D in physics here. So I didn't love him. Okay. But thank you again. Let me try to keep an eye on whether you're. You can come on up, we'll get you, because you've got one. Yours is here.
No, I just have notes. Yeah. In case I need them. I didn't know if it would, but I figured it would be better to have them. Than to just stare at a computer, trying to think of what to say.
So if you stand about when you're ready.
Oh.
So here's where you are on to the remote audience. So if you stand sort of here or along this line, you'll be visible. But whenever you're ready, go ahead.
I'm gonna make sure to turn on this pointer just in case I need it.
Yeah, and you can write from that pad. You can write from this too, but this is more responsive. Okay, good. When you're ready, go ahead.
Sure. Okay. Hello, everyone. I'm Gerald. I'm going to be presenting on my project on the identification of drivers of neuroendocrine transdifferentiation in bladder cancer. So just a quick background, my project.
Yes, sir.
Oh, nice. So my project involves small cell carcinoma of the bladder. Now this is a really rare variant of bladder cancer and it accounts for approximately 1,000% of annual bladder cancer cases. And so this means it affects about 4,000 people every year. This disease resembles neuroendocrine cell morphology and molecular features. Additionally, it's a very aggressive disease and it's often diagnosed at late stages. So this means that the cancer is often spread beyond like the walls of the bladder by the time it's diagnosed. Also, this disease has a high metastatic potential. So what this means is that it often spreads to other distant locations in the body. So some common locations that it spreads to is the lymph nodes, the brain, the lungs. And so this disease generally has poor outcomes. So if we look at this figure shows the survival probability of a cohort of patients that were diagnosed with either recurrent or metastatic small cell carcinoma of the bladder. And so if we come over here, it's the survival probability is a function of time. So after 1 year, the survival probability of this cohort dropped all the way down to 33%. So to put this in some context, the relative survival, or the 5-year relative survival rate for bladder cancer patients as a whole is 77%. So just comparing these numbers, it's significantly worse for patients diagnosed with this disease. Additionally, due to the rarity of this disease, its biological and genomic characteristics are generally unknown. And also there aren't any standard treatment protocols that have been proposed for this disease. Instead, all of the current treatment is extrapolated from that of small cell lung cancer because the diseases are generally pretty similar. Also, so there's been recent investigation into the transcriptional profile of small cell carcinoma of the bladder. And so these studies found, sorry, one step back. So this is just showing essentially which genes are being expressed in the cancer cells. So that's what the mRNA transcriptional profiles are talking about. And so these studies found a discrepancy between the histopathology. So that's just the diagnosis of cancer cells by observing them under a microscope. And then the mRNA profile of the actual cancer cells themselves. So they found that 1% of the cancer cells of the patients were diagnosed with neuroendocrine bladder cancer by the histopathology, but 5% of the tumor samples that were tested represented a neuroendocrine mRNA profile. So this implies that the disease is potentially like more prevalent than has been previously reported or previously thought. So just a quick summary, despite like the rarity of small cell carcinoma of the bladder, it is still a very aggressive disease and has limited treatment options. And also it may be more prevalent than we previously thought. So this disease is believed to result from a neuroendocrine transdifferentiation of bladder cancer cells. So this is just when cells undergo a phenotypic shift. So this is a shift in the gene expression that these cells have from the phenotype of one tissue or organ, so that of just regular bladder cancer cells, to that of another tissue or organ, so neuroendocrine cells in our case. The mechanisms behind neuroendocrine transdifferentiation are currently unknown, and a better understanding of the genetic and epigenetic drivers of this could lead to therapy specific to this disease, and that could help increase the survival of patients that are diagnosed with this. And so to investigate these drivers, we aim to develop a reporter plasmid that will provide us a quantitative measure of neuroendocrine marker proteins. And then this gave us the overall objective to use these reporter plasmids along with a Crispr screen to identify candidate genes that are instrumental to this neuroendocrine transdifferentiation. So a quick look at our reporter design. There are certain neuroendocrine marker proteins that are expressed significantly higher in neuroendocrine cancer cells when compared to regular bladder cells. So if we look at this figure just shows the expression of DLL3, which is a well-studied neuroendocrine marker protein. And so as you can see, the expression in the small cell carcinoma of the bladder tumor cells is significantly higher than that of just regular bladder cells here. And so if you have a reporter plasmid that expresses green fluorescent protein under the same promoters, so the same regulatory elements as these neuroendocrine marker proteins, cells that express these marker proteins will also express green fluorescent GFP. So if we're looking at this image, we would keep the regulatory elements upstream of the genes the same here and here. All we're changing is what is the gene that's actually being expressed. So instead of the marker proteins, for example, DLL3, we'd be expressing green fluorescent protein. And if you're looking at the cells under a microscope, they look fluorescent green. And so the promoters that we'll be using are all several well-established neuroendocrine marker proteins. So there's synaptophysin, chromogranin A, delta-like canonical Notch ligand 3, so that's DLL3 that I was talking about earlier, and then insulinoma-associated protein 1. And so to develop these reporter plasmids, we first have to use a nested PCR method to amplify the promoters. So essentially starting from the human genome and getting just the sequence that we want. So these methods first require us to increase the reaction temperature up to 90 degrees. So we start with the template DNA strand, it's double-stranded DNA. We increase it to 90 degrees, and that causes the two strands to denature and split apart into two single strands. And then we drop that temperature down to what is the characteristic annealing temperature of the primers that we're using. Now these primers are just short single-stranded DNA molecules that are complementary to locations on that template DNA strand, and they just define where the start of the elongation of the reaction begins. And they're where the enzyme DNA polymerase will bind to the molecules. And so the next step is just where the enzyme DNA polymerase binds and then starts adding on free base pairs here. And then you would just replicate the cycle of 24 to 30 times, and you get exponential growth of just that specific DNA sequence of interest. And we used a nested PCR method, which just uses two polymerase chain reactions with a set of outer and a set of inner primers, just to increase the purity and the specificity of our final DNA product. The next step after we have these promoter-like inserts isolated and amplified is to digest them with restriction enzymes. And so what restriction enzymes do is that they look for very specific DNA sequences and then make, I drew it here, an L-shaped cut in the DNA sequence. And you get these overhangs here that are complementary to each other. And these complementary overhangs, you can, if they are complementary overhangs, then they can be joined back together later on using an enzyme called DNA ligase and a ligation reaction. So if we're looking at this diagram, you start with the green part would be our insert with some extra little bits of DNA that you wouldn't really want. And then the black parts are your restriction sites. And this blue section would be the vector that we're going to be putting our insert into. So you're going to use the same restriction site on your insert and also on your vector to get just the insert in the vector. And you mix them together and ligate them to recircularize your vector. So after you have your vector isolated, you would transform it into competent bacterial cells. So these are just cells that naturally incorporate foreign DNA or uptake foreign DNA under certain environmental stresses. So heat shock, the temperature goes too high, then they're just looking for other DNA that might help them survive whatever these stresses are. And then you would just grow the DNA under a specific antibiotic. So we used ampicillin. So this is just the method to make sure that the bacteria we grow are just the ones that have the plasmid in them, because all of our vectors have an ampicillin resistance gene. And then because we use the same restriction site on both ends of the vector, it could ligate in, or both ends of the insert, it could ligate in either the forward or the reverse reaction, but only the forward reaction, or only the forward direction is useful to us. So we then have to isolate the plasmid and sequence it to make sure it's in the correct orientation. So on to our progress that we've made. We have determined the optimal conditions for the nested PCR of those promoters I was talking about earlier. The main optimization that we had to do was in that annealing temperature that I was talking about. So the annealing temperatures are not the same for all primers. It's all dependent on the actual size of them and then the base, like the nucleotide composition of each primer. So this is just a gel electrophoresis image of our primers. So this just separates DNA based off of their size. So smaller pieces of DNA will travel faster through the gel. And so this shows the different bands or primers that we have, their expected base pair size, and then also the annealing temperatures that we use for all of them. And so if we look at them, they're all a little larger than 1000 base pairs, and they line up with the 1000 band marker, just a little higher than them as expected. And then also they're of like the correct like relative height to each other. So if we look at INSM1 and CHGA, this one's slightly larger, so it's also slightly higher up on the gel. We then went through the process of digesting and then ligating these promoters into our vector, and then growing them in bacteria, but we only got positive growth in DLL3 samples. And so then we sequenced it to confirm its orientation, and we did actually observe that the DL promoter sequence was inside of our plasmid, which was good. The only thing was that it appeared that the GFP sequence was missing out of the plasmid, so we couldn't actually use it as a reported plasmid, but it did still demonstrate that this vector would uptake the insert and that it would work. We just needed to find the optimal conditions for the ligation between the inserts and our vector. And then because GFP was missing, we decided to sequence our original vector without the inserts in it, just to make sure that GFP actually was there. And so down here is the sequencing results. It is there. There must have just been something that happened with some kind of recombination from the bacteria after it was transformed. So on to future work. After we have these reporter plasmids working, we'll have to transduce them into cells with known expression of these neuroendocrine bladder cancer marker proteins to validate their activity. So they would express green fluorescent protein, as I talked earlier. This is just an image of what they would look like under a fluorescent microscope. And then after that, we could use these reporter plasmids in conjunction with a Crispr screen to identify candidate genes that is necessary for neuroendocrine transdifferentiation. So this Crispr screen would use a library of short guide RNAs. So that would be this little red section of DNA here. And these short guide RNAs would be complementary to genes in the human genome that we would want to like turn off or knock out. And so these short guide RNAs would complex with a Cas9 protein. And then once it finds that specific part of DNA, it's going to make a double-stranded cut. And then cells are going to try to repair this double-stranded cut. But mutations will usually occur, and like the repairs are not always perfect. So that's what causes the knockout of that specific gene in the cells. And so after specific genes would be knocked out, we would measure the expression of GFP in those specific cells to see how that knockout affects the GFP expression, and then in turn the expression of these marker proteins. And so the way that we actually get all of this genetic material into eukaryotic cells is with lentiviruses. So one example of a lentivirus that you guys have probably heard of is HIV. We're not actually using HIV, but that's just an example. And that's just because these viruses incorporate their genome into eukaryotic cells. So and then other future work that we could do is we could exchange the green fluorescent protein in the reporter plasmids with a red fluorescent protein. So that way we could use the two different reporter plasmids simultaneously in the same cell and observe how one gene could affect like the expression of both of the two marker proteins at the same time. And that would just increase the fidelity of our model. Thank you. Any questions?
Any questions? The remote audience can just unmute yourself if you have it. Otherwise, Alice, okay.
I saw you was trying to mix in the green fluorescent protein into the one plasmid. And how would you select the one that failed to mix and filter them out? How would you select the one that failed to mix with the protein, the green fluorescent protein? And just transduce the ones that you want into the genes.
I'm not sure if I fully.
I mean, when you are making plasmid, there are some plasmid that you failed to make. How
Yeah.
would you select and filter them out?
So I think that step. So normally the way that we're looking to filter out the plasmids that we don't necessarily want, or the bacteria that we don't want, involves that ampicillin resistance gene. So if the bacteria doesn't have our plasmid inside of it, it doesn't have the gene that that confers ampicillin resistance. So it dies in our agar plates. But for this case, I think it was just that section of like GFP that the bacteria have certain enzymes that can like recombinate certain sections of viral like DNA. And so I think it was something specific that happened just with that. And I'm not 100% certain if there's anything we can do prior to having sequenced it to know that that had occurred. Because if it still has the plasmid with just the ampicillin resistance gene, but not GFP, it'll still grow. There might be like background expression of green fluorescent protein in cells. So we could look at them under a fluorescent microscope and then mark colonies, like bacterial colonies that fluoresce green under the microscope. And that might be a method to do that.
Thank you.
So if no one else has a question, I have just one simple one, I think. When you got to future work, you were using phrases like we will or we plan. I'm curious if you plan to continue doing some of this for any sort of extended time.
Yeah, I actually was accepted into like a cancer case summer research program or fellowship, and I'm planning on continuing my work with Dr. Meehan like throughout the summer with that.
Okay.
The base.
That's interesting. It doesn't happen very often. Okay, unless there are any other questions though, so applaud your own once more. Oh, it's my screen share. And we'll see if our other speakers are standing out in the hall. One more here, okay. I think you may have a sandwich in the fridge waiting for you. Should I open it? What, you're using your own laptop?
Yes.
Remember, whenever they're done, while they're saying, go ahead and set up. Connect to the Zoom, the information's there if you need it. When you leave, there are some cookies and other things around. Be best if those left with you.
Thank you.
Okay, otherwise, somebody's gonna have to call in the grad students. Wait, call them the locusts. And when you speak, it would be best to speak to around here because that's when you'll see this display will pick you up if you're over sort of anywhere on this line.
Okay.
So I can start over a bit more.
And so we log into a Zoom call and
right there.
Yep.
Professor Taylor is doing dual Zoom meetings, I think. After the Senate and this. And I'd like to.
Yes, I'm double zooming.
Thank you.
It'd be best if you use the PowerPoint tool built in. Are you using PowerPoint?
No, we're using PDF.
Yeah.
I don't know if it has a pointer built in or not, because no one will be able to see that thing remote.
Oh, yeah. Ooh, yeah, I don't think there is a pointer, but we don't really have any graphs that we need to point specifically.
We just use the mouse if we really need to. Meeting ID, can you read it out to me?
937.
Sure.
354 1772. Password is all caps physics. You could erase the DNA sequence behind you in case you need the board.
Oh, yeah, I mean, I don't think we will, but I just want just in case.
Yeah, the remote audience can't see the board.
Right, they can't see the board. All right, I'm just gonna. Do we want video on or no?
Let's share your screen then.
I guess we'll probably, yeah, it's more important to share the screen. So I'd probably share this one then.
Yeah.
But we don't really want the browser to be visible. We want it more like on full screen.
Well, that's what I'm gonna do.
Okay. There we go. Perfect.
No, I apologize. I made an error in the timing. I only gave Gerald 10 minutes. That's why you're starting a little late.
Oh, okay.
That's why I'm getting cataract surgery after commencement.
All right,
You have to turn your speaker off because it's using.
it's using that?
Yep.
Okay, good.
Yeah.
Then the remote people can speak through that.
Then we should probably get rid of, well, can I, is this a touchscreen?
Yeah, it is. Was. I haven't tried to play with it. I know it's annoying. I was worried about something going on. When you're ready, but you may want to like stand here, Professor Taylor won't be able to see you.
Where's
You
the
can see-
camera
You can see
at?
here.
Oh,
You can see on
yeah,
this
yeah,
display
I can
where
see.
the angle is coming down.
Oh, it's that one.
Great, yeah. Maybe I could, can I like, I don't want to like, I don't want to mess with that.
Mm.
All right, are you ready?
Yeah, ready. Let's go.
Yeah, so this is me and Christian's capstone project. And it's titled Quantum Money. We were advised by Dr. Cyrus Taylor. And yeah, I guess we'll just go to the first slide. Or do you know the first slides?
No, you literally just press arrow.
All right, well, before we talk about what quantum money is, we have to talk about what makes good money. And a lot of people really don't think about this, but money really has a few very sort of key characteristics that make it a useful currency. And what makes good money is me and Christian boiled it down to sort of three main things that we wanted to focus on in the project, which was that money is hard to copy, good money is hard to steal, and it's easy to spend, or it's very fungible, right? And classical money, the money that we have in bills, or more recently, sort of the money that is stored digitally on credit cards and stored in banks and collateralized by the reputation of those banks, is all three of these things, at least to some extent, right? So it's very hard to copy money by counterfeiting it, because there's all sorts of measures that we take to the serial numbers with the minting process, with the Federal Reserve, and large government agencies that keep track of a great many number of records to make sure that money that is circulating around has not been counterfeited. That money's relatively hard to steal. You can put your money into a safe. You can store your money in a bank. You can put it in a safe deposit box in that bank. You can store it on a credit card. And a lot of times those credit cards have policies where someone steals it, you can cancel it, and get your money back. And then money is easy to spend. You can go somewhere and people commonly accept your money. You can pull it right out of your wallet, or you can swipe a credit card. And these are really the three components that make classical money or any currency really, really good. And the classical money that we use today can do all three of these things to some extent. So,
Cryptocurrency sort of uses cryptographic algorithms to emulate those properties that we want in a currency. So the main way that it's done nowadays with cryptocurrency is by using like a backbone called a blockchain, mining and appending algorithms. So a means of generating and adding on currency to the overall tally of the currency circulating in the world, and by digital signatures, which are used to ensure that people sending you these digital assets are trustworthy, which is something that's kind of already built into the currency that we see nowadays, but is a little bit more explicit in the cryptocurrency scheme.
Yeah, and one thing I'll add is that what cryptocurrency, which we've seen just explode in the last few years, really sort of solves compared to that last slide with classical money, is that now you don't have to have some kind of centralized agency such as a government or a bank that is having to be trusted in order to mint the money. Now the security of the money is inherent to the blockchain, and the security of that blockchain is inherent to the security of the hard to solve cryptographic algorithms and hashes that are being used to generate new symmetric keys that people are using for their digital signatures.
So cryptographic hash functions are essentially ways of that cryptocurrencies add on money to the current list. So basically, these hash functions take some sort of string, text, number, and spits out a string of constant of length to the end. And the kicker is that any small change, L naught, any difference between those two, when you feed those through this hash function, you will get vastly different outputs from the function. And what you can do is you can basically say, all right, if I take the previous block that has the tally of all the cryptocurrency in the world, and I feed this through the hash function, what do I have to add on to that block such that the result will start with like 30 zeros in a row? And the first person to get that in the world is usually is awarded that coin, and the proof of that is attached onto the block, which is then redistributed.
And the really important thing about hash functions is that something that me and Christian will talk about when we motivate our quantum cryptocurrency is that it's an example of what's called a one-way function. And a one-way function is something that you can put an input into and it gives an output. And it's very, very easy to check that that output satisfies that function, but it's very, very difficult to take that output and find the original inputs. So with the blockchain, which is this decentralized ledger, a great many people who are part of the entire currency can check that someone's coin is legitimate by using the keys that they generated, putting it back through the hash function and getting the same result, but they can never find out what those people originally used to seed those keys. And so that's an example of a one-way function, and it's a real central part to how me and Christian are gonna motivate our quantum currency. So I'll talk a little bit here about digital signatures. So a digital signature, I guess the best way to describe it is by talking about what a classical signature is. So if you think about what you're doing when you write your signature on a piece of paper, is everyone has a very specific way that they write their signature, right? I might write my name with a cursive N, a little tally on the A, and a loop on the H. And that's something that I can do very, very easily with muscle memory. And someone else can check it very, very efficiently, very easily and say, oh, that's Noah Linzel's signature. I can check it against many other pictures of his signature. But it's very, very difficult for someone to then, even if they have many images of my signature, to write it the same way, because their muscle memory is not going to be perfect. And there's going to be ways to tell that it doesn't work. So a signature that you write on paper is actually a weird example of a one-way function. It's very, very easy for me to write my signature. It's very, very easy for anyone else to check, but it's very, very hard for someone to perfectly forge my signature in a way that's not discoverable. And so digital signatures are one of the nuances in which we sign the blocks in a blockchain with which we create new cryptographic coins. A person has their own unique digital signature that they seed with random keys. Oh, I think I just have to move the mouse every now and then. That they seed with random keys, and that can create their own unique digital signature that is easy, again, for everyone else in the network to check. I don't know why it's doing that.
You're still on the Zoom. So yeah, we're still on the Zoom.
Okay, but I want the people in here to be able to see as well.
Yeah.
Oh, well. It's very, very easy for other people in the network to check it. And this will be another very important component of how me and Christian motivate a quantum digital signature currency. And me and Christian have already talked about blockchain. I don't think we have a whole lot to discuss here, but like, again, it's nothing more than this distributed public list subdivided into blocks where each block contains only 3 data. The cryptographic hash, the previous block, a timestamp, and the transaction data, i.e. like how much was exchanged. And this is the backbone upon which cryptocurrencies are created. So Christian and I are both going to talk in this slide a little bit about, well, why do we want to integrate quantum information? Well, it's a really sort of natural step when you take into account a lot of the very unique characteristics of quantum information. There are phenomena that we find in nature, such as nuclear decay, a lot of phenomena having to do with electron spins that can produce truly random processes that we can use to see truly, truly random number generation. Up until now, all random number generators are digital and they all have periods to them, which means that in some sense, they are actually not random truly, and that means that they're compromisable. And then the no cloning theorem ensures us that no unknown quantum state can ever be copied. Do you want to add something?
Yeah. With the quantum randomness, you can imagine if you have some sort of qubit that's very highly entangled to the environment around it, you can imagine that being super noisy and taking some sort of measurement. would yield about as random numbers as you can get. And that is illustrated in the fourth bullet point. And yeah, so the no cloning theorem says you can't just take a quantum state and copy it without making some sort of other compromises. And there are properties of quantum states and quantum systems that are that make it powerful for information processing and random number generation.
So here's what me and Christian have done, and this is where we're going to sort of transition between introducing you guys to the concepts that we've motivated and what we've actually accomplished in our project. So what me and Christian have done is we have used Python. We have used a great many of different packages from different companies such as IBM Quantum, Argon's sequence platform, which I'll talk about a little bit more, as well as a couple of proprietary packages that are given to us by labs around the world. And what we've done is we've programmed a protocol that simulates an acid exchange network that is backed by quantum random processes. So our simulation, we can encode these any message that we want, or a coin, if you'd like, into a quantum state. We can simulate that quantum state and send it over a realistically simulated quantum channel secured by a quantum key distribution, which is something that we'll also talk a little bit more about. And these messages are signed using a real digital signature protocol, a quantum digital signature protocol, because the one-way function that we use actually acts on quantum states on the block sphere. And we'll represent that as well. Christian's made a couple of very good figures with that. So those are the three components of what we've done. And now we're going to talk a little bit about the packages that we've used to do that, the motivation behind it.
Yeah, so this is just an extension of that last slide. Simulated quantum channels, QKD, and the quantum digital signature. The one that we focused on the most and is the most solidly produced so far is the digital signatures. The QKD scheme, quantum key distribution, involves a Python package called Sequence. So it's a simulator, and we got that to its earliest stages, but we're going to talk about how it can be improved upon later on. And so what QKD does is it provides transmission security. It establishes a channel where two parties know that they can be sure that if there's someone interfering, that they can just cancel and stop transmission. That's established by QKD. And what the digital signature does is it verifies that this person who is sending the message is indeed that person. And you have to be able to make that distinction between identity and transmission security. And we will now provide some of the work that we did.
And so in principle, if you have a network where the transmission of your message is secure, i.e. no one can intercept it and gain useful information from it, and then also the identity of that information is secure, i.e. I know because of the digital signatures who I'm getting it from, then theoretically that network is now an informationally secure network. And so nothing about the information that is being transmitted over that network or the information about the person that is transmitting that over that network can be compromised. And because we are using quantum information, quantum random numbers, and quantum protocols here, this whole scheme is not just secure in practice because it's very, very hard to break or you need a big computer. It's secure by the laws of nature itself, because quantum information is not compromisable when it's used in these schemes. And we're going to talk a little bit more about how we realize that in code. So here we just sort of run through our relevant software. Sequence is an open source platform that's being made by Argonne National Laboratory in collaboration with HRL Labs, which I'm actually working at HRL right now. So I've gotten to work a lot with Sequence. And it allows for a realistic and scalable simulation of a very large long distance quantum network. The ANU QRNG, I'll let you talk about Qiskit a little bit more. I think ANU QRNG is a package that has been provided by Australian National University. And they seed their quantum random numbers in real time in a lab by measuring the quantum fluctuations inside of a vacuum chamber. And they, in real time, upload those results to a server. And that's what we use to seed our random numbers. So the random numbers in our protocol are truly quantum random numbers. And then also we used IBM Qiskit, which I guess, yeah.
Yeah, so Qiskit is what we've been able to do the most with. We've had trouble making Qiskit and sequence talk to each other nicely, but we're confident that they can with more deep in-depth investigation and with more like look at how to actually build the software that can talk to each other. But Qiskit is a platform via IBM where basically you can build quantum circuits, implement logic gates, and produce some really nice and pretty graphs and figures of the resulting states. And you can even incorporate some, you can even send these jobs to either simulator or to an actual quantum computer by IBM. And so that's a really cool package to have.
This is an image of the lab at Australian National University, just to show that we are actually using a real laboratory setup that's on the other side of the planet, and that is reporting quantum random numbers to us. We had to do a little bit of weird imports with the packages because it's a very small team. So the packages that they had were kind of weird to implement to our code. But we are indeed actually seeding in real time measurements that are coming from the vacuum chamber right there.
So imagine qubits highly entangled to the environment around them, represented by a very non-sparse density matrix. That's kind of what the process is utilized in that experiment. So quantum digital signatures, we are using the 2001 Gottesman-Schwang protocol, which is the most simple and rudimentary one, but it works and it produces results. Basically, the way it works is Alice and Bob. Alice wants to send a message to Bob. She basically takes her message, hello world, and for each one of those letters in hello world is encoded into its a 7-digit binary string. So for each letter, you have 7 digits. And for each one of those digits, she produces her classical keys and which are fed through quantum one-way function to produce quantum states.
Shown at the bottom right there.
That's the one-way function, psi. And the quantum states and the classical keys are sent to Bob, who also knows psi. He takes these classical states, feeds them again to the one-way function, and compares the quantum states that he received from Alice and the quantum states that he produced himself. And if enough of them match, he accepts the message as transmissible and from Alice. That's the simplest protocol that we can find, and the one that we were able to actually produce, as you'll see on Python.
And so I just want to also add that because Alice is signing every single one of the bits in her message with that quantum one-way function, then Bob can check for every single bit of the message, did this bit come from Alice and only Alice? So not even one bit of the message could be flipped without Bob realizing that there's a problem. And so that makes the message transmission secure. And our QbS algorithm.
That's precisely what I said.
Yeah, this is exactly what Christian ran through. But this is just, she's producing these keys, she's sending them to Bob, and Bob is checking them with the quantum one-way function.
The security parameter M is this protocol is exponentially secure in M. So the larger M is, then the more classical keys she'll produce per bit, and so the more Bob can check whether he can basically set the threshold to be higher.
And yeah, Kristen's going to run us through a little bit of our code, and I know that we're short on time, so we're probably going to just sort of buzz right through it. But
I will show what this looks like.
Yeah, so I'll give a little bit of background on what Christian's about to show, which is basically Christian's about to show what Bob is going to be receiving from Alice every single time she transmits a message. And so what you're going to be seeing here is what we call the packet. So the packet that Alice is sending to Bob has to include two things. It has to include every single bit of every single letter of the message that Alice is sending to Bob. We use examples such as hello world or little messages like that, but this is where you put in the serial number of the quantum coin that you're sending over a network. And then with every single one of those bits, Bob is receiving Alice's public and private keys. And Bob can then use that with the quantum one-way function to verify that this message is indeed coming from Alice and that you have a secure channel that hasn't been compromised. Yeah. And so that's this entire packet that Bob is receiving. And when he receives it, he will then check it by comparing the results of what he's expecting to the results of what he gets. And if they come within a certain threshold of each other, depending on Bob's risk tolerance, he knows that he's getting a message from Alice and Alice only.
Yeah, so this is the encoding of the message H with security parameter 3 and key length 4. And so you can see this is the first bit, this is the second bit, the third bit's a 0, the fourth bit's a 1, the fifth bit's a 0, the sixth bit's a 0, and the seventh bit is a 0. So
Do you want to explain what the security parameter 3 is, like what that means?
what was that?
The security parameter 3.
It just means that the security parameter is M. The security parameter is M. So that's the amount of basically for each bit, the amount of keys that Alice produces.
Yeah. And so obviously the bandwidth of this protocol is going to be really, really low because every single bit of information has to be signed with this massive amount of data. So our objective here, and something that me and Christian always joke about was that this was a physics project, not a computer science project. We weren't trying to make this the fastest, most efficient protocol. What we were trying to see is could we use quantum information to secure an asset exchange network? Whether or not it would work really, really well in practice, I don't think that you could do this on a classical computer with useful sizes of coins. It would probably have to be run on a cluster, but we wanted to make sure that we could prove that it worked in practice.
So we will proceed onwards. Yeah, there you go. Yeah. And then I'm gonna make a full screen. Great.
Yeah, so also, we include a few screenshots. This is how we're producing Alice's original keys. This is very, very simple code right here, but you can see where we've imported the QRandom ANU QRNG lab that's feeding in our quantum random numbers, and then we're feeding that through a hash string function that is then producing and integer converting the bits that Alice is using for her keys, and those are the pairs right there. This is just code that we didn't get to see in the demo because it's kind of running behind the scenes.
It's hidden in there.
Yeah. And then our one-way function, again, this was some of the most simple Python of the project. The really, really complicated part was compiling the data into those packets for Bob. But the quantum OOF, as we call it, the quantum one-way function, is implemented with this code here. You can see that we're just taking these digits and then we're returning that function psi of omega that you guys saw on the slide earlier, that Bob can check very, very easily, but it's extremely difficult for somebody to invert that function.
Here's a few examples. I know I said earlier that Qiskit can produce some really wonderful diagrams, and here's a few mappings of W equals messages equals 001, 101, and 011, feeding them through the one-way function and then plotting their resulting quantum states on the block sphere.
Yeah. And Professor Matur actually pointed out at one of our mid-semester meetings that there's actually no reason that we have to be dealing in a two-dimensional Hilbert space here. We could be dealing in an n-dimensional Hilbert space, but you can't represent that in a sphere. I mean, you probably need like an n-dimensional hypersphere to represent that, so you can't do that on the screen. But there's no reason that our one-way function couldn't be of a much higher dimension and therefore even much more unbelievably difficult to invert. But the reason that we use the Godesman and Chuang one is because it provides these very elegant examples. And in practice for the project, it made the most sense for me to use that one. Okay. Yeah, so looking forward, our simulation is really modular right now. So we have, we've got QKD working. We've got those packets to Bob working. We've got the sequence packages imported, and we've been running simulations on quantum networks. But we don't have this one big unified block of code that says send coin to Bob from Alice, mint the coin, and do it all in one step. And that was something that me and Christian didn't have time to do. But again, we talked about computer science versus physics. It would have been really, really cool to make this entire app, this platform that does this. But the focus of me and Christian's project was more to understand the nuances of the physics and the implications of using quantum information. But that would definitely be something cool to do further. You want to also add anything?
Yeah, and we aren't really sure what the actual asset looks like. Right now, our quantum digital signature sort of it can send, our example was the letter H. So what will the actual how can we make that so that well, first off, what is an asset we can make so that it plays nicely with the digital signature? Can we tune those two up? And that'll be one of the more like complicated and enriching things about this. And we would also like to, like I mentioned earlier, make sequence talk a little bit nicer to Qiskit, because right now we have Qiskit and we have sequence, but they're not talking. So let's make them talk, and that will give us the power of factoring in some real like almost experimental-ish data with transmission fidelity, lossy signal, all of that kind of stuff that engineers care about. Being able to incorporate that into our project as well.
Yeah, we want to be very straightforward and say that we set out to make a quantum cryptocurrency, but what we ended up creating, I think, is best described as an encrypted asset exchange network. So right now, the thing that we're missing is really how is the best way to construct the coins. We've got the entire foundation to transmit those coins securely, but what me and Christian were ultimately unable to do was actually create the asset that you're sending. Whether or not that would be seeded by classical sort of serial numbers, or if we'd actually be using a quantum like fiber optic network to send polarization qubits that are encoding the coins is a question that's still up for debate. So
this was sequence. This was our this was just a proof of concept that QKD works on sequence. You can establish secret keys that two parties share. And like I said, we want to make that talk nicely to Qiskit.
And then this is
yep, and basically this this figure is it comes from the Bitcoin white paper, which has been a really big source for our knowledge on how a blockchain works, on how cryptocurrency works, and we'd like to, in the most ideal situation, to basically emulate these blocks and those algorithms, but with quantum in front of all those words. And that's sort of the gist of this project. Yeah.
And then, yeah, really quickly, we have to make the disclaimer, which is that in mid-April, like 2 or 3 weeks ago, something that was both awesome and also a little disappointing happened, which is that Christian was sort of browsing through GitHub, trying to see any similar projects to what we've done. And he found an MIT group of, I think it was about 6 or 7 students that for their Q Hackers event, which is this quantum coding event that's hosted by MIT once a year, they basically did the exact same project that we did. They used digital signatures. They even used the same quantum only function.
The same one.
They had the exact same, I mean, their code, some of the ways that they named their functions were also very similar to ours, but it had come out a year earlier. So obviously we knew that our projects didn't intersect at all. But this was really cool because one, I mean, I guess, as a couple of our mentors have pointed out, it really gave some sort of assurance to the motivation of our project. Hey, other people are thinking this too. This is a much bigger team, and they had this dedicated event, and they created something very similar to us. So we felt very encouraged by that, but also we were a little sad that someone else had thought of the exact same thing. But ultimately, we have to disclaim that here because in the end, we did end up looking at their project, and even though it's only been about 2 weeks. We definitely went in there and we cross-checked some of the stuff that we did with theirs and tried to get some new ideas.
It was validating, but also like one of those things, just like, wow, you know, they did the whole thing.
Yeah.
And they even went beyond in their GitHub code, and they actually were able to produce sort of the blockchain, or at least like a very primitive version of a blockchain from it. These guys were, it was a big team, at least 5 people, including physicists and software developers. So it was validating, kind of disappointing, but
mostly validating.
I think it's mostly validating. And with that, that's it.
Yeah. Thank you.
Here or remotely, I'm looking for hands up. So I actually have one. I think whenever you gave a progress report, I asked a question that now may have been stupid. It was about your random number generator you're using. So couldn't you just get a file of the random numbers from them? But that means it's no longer secure because anybody could copy the file.
It's cooler to do it the other way.
Yeah, it's cooler to do it the other way, which is your answer.
But
now my question is, would everyone who used this need their own independent quantum random number generator to maintain security?
So yeah, so we actually on ANU's website, they talk about that. They talk about, well, hey, you know, we're using quantum random numbers, but because those random numbers are then being stored in hard drives and computers that deal in binary transistors, doesn't that convert that quantum information into classical information? And they actually bring up a lot of really, really much more fundamental sort of stuff about quantum security and quantum mechanics than we were expecting. But yeah, they bring up that point a lot, which is like, wouldn't you, if you really wanted absolutely true, pure quantum random numbers, technically you would actually need a fiber optic cable running from that vacuum chamber into your CPU. And that would make it like, that's like maximum level security. But I think for the purposes of our project, we wanted to show that something could work in theory. And so using the quantum random numbers was just as much of a symbolic thing as it was a technological.
No, I understand that. I was just curious whether, and I wasn't worried about, oh, it's being stored digitally. I'm just comfortable. Once it's stored in any form, somebody could copy it.
Yeah.
And so then you would, you would break your security.
And me and Christian also thought a little bit ahead and thought, well, if this became like, if you wanted to really create this decentralized quantum asset network, everyone would probably want to have a little box in their house underneath their desk that had this little quantum vacuum, and as well as a little cylindrical like rubidium quantum memory to store your coins in. And you know, it's just really like, I mean, it's kind of like, I don't know, it's fantasy stuff, but it's
well, right next to your quantum computer.
Yeah, right next to your
quantum computer. Actually, just be an app on your quantum computer.
Oh, sorry, did someone have a question?
Well, Professor Brown has his hands up.
Brown.
So, well, this was amazing, just absolutely amazing. You guys are great. You know, the big thing, you know, and jumping ahead, the big thing that everybody's talking about is energy, the huge problems of energy with cryptocurrencies and things. Is there anything that this direction has to say to speak to that problem?
Yeah, well, one thing I mean, did you want to add anything first?
Go ahead.
One thing I can say is that as far as I know, I mean, me and Christian learned so much about just cryptocurrencies in general when we were doing this project. And one thing I've realized that leads to a lot of the energy loss in classical cryptocurrency schemes is you're constantly having to make your hash functions longer and longer and longer. And you're constantly having to then therefore use much more powerful clusters to solve the hash function problems to then mine a new coin, because you can't recycle the same hash. But one idea that I had, and I've talked to Christian a little bit about this before, is, well, technically, if you have entangled quantum states, right? When you're thinking about a Hilbert space of quantum information, 90% of that Hilbert space, if you've got multiple entities interacting, is made up of entangled states that you can't access classically. So if you're accessing this much bigger Hilbert space, then you can actually do this thing called entanglement recycling, which is where you can actually use the same quantum hash that you're using before to make another quantum coin, if you will. You're not having to make a new one. And like I said earlier, the reason that cryptocurrencies, especially Bitcoin, I think last year used the same amount of energy as like all of this country of Kazakhstan or something ridiculous like that, is because now you've got all these PlayStation GPUs in the middle of the desert that are running constantly, producing all this heat, because they're having to constantly solve harder and harder and harder hash problems.
I have a real burger?
No, I already ordered it.
Oh, I'm sorry.
It's better.
So the question is, can you buy a real burger with these currencies?
Yeah, your question is very interesting. It's not something that me and Christian really explored a lot. I don't know if you have anything to say.
What about the energy or? Yeah. So that's something that we were thinking. Yeah, the energy is something we were thinking about like at the very beginning of the project. It's just not really where it went. I think it's looking into like what could possibly like the hardware efficiency of like quantum computers that would be needed to implement the algorithms that we've shown you guys, is something that's worth like looking into, and could be its own project, like standalone project. That's just not really where we went. I think that any energy sort of like trade-off or any sort of like efficiency gain that could be done with the algorithms we proposed is probably almost certainly like some ways off. Like I don't really see it any really like energy improvements in the near term. But like looking forward, if quantum computers really are all they're cut out to be, and like the hardware is developed like with, along with the hype that people are kind of projecting, then we might be able to see in the future like a new sort of more efficient way of doing cryptocurrency. But yeah, I think that it's kind of insane that we just have it's a great idea in theory to have a blockchain that has the whole history of the entire chain built, everyone in the network has it. But the problem with that is you're solving these ridiculously hard problems every time a new coin is mined. And then you have what are you going to do when the total ledger on everyone's computer is more than like a couple terabytes? It's going to get huge. And those are fundamental problems with cryptocurrencies, along with the energy that may be able to be worked around by quantum. That's something that is definitely worthy of its own project or projects in the future.
Any other questions?
Thank you.
Thank you.
In order to leave, you must take a willing cupcake. A cookie or whatever. If somebody wants the whole tray, go ahead and take it.
Thank you, Christian and Noah.
Of course.
Thank you.
Well done.
Thank you.